set off alarm in your mind, the idea that it's a nucleic acid and that's one of our organic compounds. All right, and this is actually the genetic information. The real purpose of the genetic information is that it's actually supposed to be instructing your cells how to build proteins. So we always think about genetic information as really being about traits and how you look and all of that information, but really what it is that genetic information is responsible for creating proteins, and those proteins are what are responsible for your traits. So you've typically been missing a step in there. All these different proteins that are created have a wide variety of functions. So for example, we have enzymes, and we should know that enzymes are going to be controlling metabolic reactions. So enzymes for metabolic reactions. We have proteins, excuse me, proteins that are going to play a role in structure. So for example, we use proteins for building muscle and connective tissue. We have other proteins that play roles in protection. For example, antibodies. Antibodies are a specific type of protein that fight off infection in your immune system. And I just want to remind you guys that when we talk about proteins, we need to continue to remember that proteins are made of amino acids. So chains of amino acids. The amino acids are the monomers of the polymer protein. And there's a bunch of other functions, but these are three pretty good examples for us to really focus on. The structure of DNA is that it's referred to as being a double helix, double meaning two, helix being that kind of spiraling shape. Sometimes it's called a twisted ladder, a spiral staircase. And basically what it is two chains of nucleotides. So you're going to have two chains. You'll have phosphate, sugar, and a base bonded to another sugar that's bonded to phosphate, etc., 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 etc. Same thing on this side. Da, 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 da. But the interesting thing is this anti-parallel. And this is something that we didn't really focus on in biology. But if you look over here at this picture, you'll notice that one side seems to be right side up, where the phosphates are above the sugar molecule, and the base still comes off the sugar. But on the other side, they appear to almost be upside down, on the idea that they're going in an opposite direction. That's that anti-parallel, and that's actually how it really does run, that they run one in the opposite direction of the other. So talking about the actual structure of DNA continuing, we have that sugar-phosphate backbone, the idea that we have sugar, phosphate, and the bases are in the middle, but the backbone down the side is always the same, sugars and phosphates. And the sugar is deoxyribose, which is where DNA gets part of its name from. Deoxyribo is named from that sugar. And like I said, the bases are attached to the sugars. There are actually hydrogen bonds that keep the complementary base pairs, and we should know complementary base pairs in DNA, adenine with thymine, guanine with cytosine. And interesting thing is that in each of these cases, what you're having is two rings, like in the case of adenine and in the case of guanine, that are going to bond with a single ring. So the structure of these bases is two rings versus one ring, and so long as they pair with the opposite or the complement, your rungs of your ladder will always be 3 rings wide. Our adenine and guanine are those 2 rings that we refer to as purines. And thymine and cytosine are referred to as being pyrimidines. All right, so that's kind of what keeps the consistency the same. And if you look up here, it's kind of difficult to see, but what you have is a double ring bonded to a single ring, or a double ring over here to a single ring, single, double, double, single, and so on. They just kind of look a little different. It's kind of hard to see in that picture. Also, it's important to point out that that base sequence is really what the genetic code is. And keeping in mind, again, that the genetic code is not about your physical traits. It's really about turning that base sequence in DNA into a chain of amino acids or into a protein, and that protein being what's responsible for giving you your traits. Also, when we look at DNA, DNA coils around histones. Histones are a specific type of protein, all right, and these histones tightly wrapped up with DNA is what we call chromatin, right? That's that genetic material that's in the nucleus. So if we look down here, right, we've got our strand of DNA, that's right here. Sorry, I lost my pointer for a second. Our strand of DNA, and then here are those histone proteins, and the DNA starts to kind of wrap itself around them. And if you see, that's when we refer to it as being chromatin, when it's kind of coiling around and wrapping up around those proteins. DNA is also arranged in genes, and what that basically means is that that's a chunk of genetic information, a segment of the DNA for one particular protein. So we can kind of look at it as being like over here. This chunk right here is a segment of the DNA molecule, and that segment would be referred to as being a gene, and that gene would be responsible for one particular protein. And ultimately, the DNA is what holds your full set of genes, all of your information, all of your genetic material. It's important to also point out the fact that each cell needs a full set of genetic information, needs a full set of DNA, and the reason for that is that each cell needs to be able to create its own proteins for metabolism or that are needed for other processes in the cell. So it's important that each cell has its own set. In order to accomplish that, a process called DNA replication occurs. And I think it's pretty simple to understand, replicating to copy. So it's copying the DNA. And we're pointing out that it occurs in interphase, which shouldn't be a surprise. Interphase is cell division, and specifically it occurs in the synthesis. Sorry guys. In the synthesis phase of cell division. So in the synthesis phase of interphase of cell division. When we look at DNA replication, what's actually going to happen is that hydrogen bonds, right, are going to break. They're actually going to start to split between the complementary base pairs. So it's almost like the DNA molecule unzips and unwinds. Right? It's going to uncoil and it's going to pull apart, just like I said there. And I don't know if you guys really remember, but last year in biology we specifically talked about a particular enzyme that was responsible for this portion of DNA replication, and we called it DNA helicase. Right? DNA helicase is the enzyme. ACE, remember, is going to be the enzyme that is responsible for this portion of DNA replication. Then what's going to happen is that the new nucleotides are going to pair up. So as we can see here, we're starting to get some new nucleotides matching into place with their complements, forming new hydrogen bonds between the portion of the old strand and new nucleotides. And again, last year we talked about an enzyme that was responsible for carrying out this portion of the process, and that was DNA polymerase. Remember, polymer, all right, is multiple molecules kind of joined together. And that's going to continue to happen until ultimately what you have is two complete DNA molecules that are absolutely identical. Right? So the results are identical genetic information, and each new molecule is half old, half new. One strand of the double helix is the original, and one strand is complementary, giving you your double helix. And it's in both of the molecules that end up the final product. A little bit more about talking about the genetic code is that what happens is that amino acids are coded for by DNA triplets. There are basically 3 nucleotides, or what we really want to focus on is the idea that it's a 3-base sequence. So DNA is read in groups of 3, and those groups of 3 are responsible for 1 amino acid each. And there are 20 different amino acids. So the idea that CGT would represent one amino acid, but GCA would represent another. So as we can see here, if we split our DNA sequence into groups of 3s, we can see that each triplet is responsible for one amino acid, and those abbreviations are for the amino acid names. Now, the thing to figure out is exactly how that genetic information in DNA gets out. DNA is confined to the nucleus, so how is it going to get that genetic information into the cytoplasm and out to the ribosomes where protein synthesis is going to occur? And that's where RNA comes in place. All right, RNA needs to actually play a role here. Another little addition is the idea that these triplets provide stop and start signals for the whole entire process. Right, so we've got starts and stops as well. So that RNA, ribonucleic acid, right? The sugar this time is ribose, which is in the name, ribonucleic acid. So we still have a sugar, phosphate, sugar, phosphate, sugar, phosphate, etc., the backbone. And we still have nitrogenous bases that come off of the sugars, but there's no complements, no complementary base pairing. And you should also recognize the fact that there's a new base in place, and that's uracil. Uracil replaces thymine in RNA. So you've got U's wherever a T would have been. That's one of the big distinguishing features between RNA and DNA. RNA is only one single strand, DNA is two. RNA has ribose, DNA has deoxyribose. RNA has uracil, DNA has thymine. Right? There's some distinguishing features that set these two nucleic acids apart, because both actually play a role in your genetic information and in protein synthesis, carrying out that genetic information. So if we look here, this is just basically showing you that RNA is built off of a template of DNA. They're actually complementing one another. So RNA is built from DNA sequence. That's how we get RNA. There are 3 different types of RNA that exist, and those 3 different types are mRNA, tRNA, and rRNA. mRNA is messenger RNA. tRNA is transfer RNA. Sorry about that, guys. Is transfer RNA, and rRNA is ribosomal RNA. So what actually happens is that mRNA, the messenger, is what's responsible for getting the genetic information found in DNA from the DNA molecule within the nucleus, out of the nucleus, into the cytoplasm, and to the ribosomes. And that happens in a process called transcription that we'll focus on a little bit in a minute. Transfer RNA is responsible for basically transferring that genetic information into an amino acid sequence. And it does that by a process called translation. Then there's ribosomal RNA that's really just a structure. Ribosomal RNA is actually part of the ribosomes. So you may remember that when we talked about ribosomes, we talked about the fact that ribosomes were made of RNA and proteins, and the particular type of RNA is ribosomal RNA. At that time, it may have seemed a little weird to you.